Great. So welcome everyone to the first NISS-Merck meetup of 2020. It's going to be on adaptive trials, and we have a great distinguished set of panelists to give you some great information on adaptive trials. I'm going to start by sharing my screen. All right. So many of you may notice that we're using Zoom software this time. We've gone to that because NISS has had great success in the past using Zoom software. So there was really nothing wrong with the WebEx, but the Zoom allows us to show video a little more easily of the presenters, and we thought everyone would enjoy that. As with previous meetups, your audio line is on mute, so you won't be able to actually interact that way. But if you want to ask questions, there's a Q&A button. If you just scroll across the bottom of your screen, type your questions in there, and we will address those possibly as the meetup goes on, but most likely at the end, I'll try to accumulate as many questions as I can and ask them of the presenters. Feel free to use the chat feature to communicate with the other attendees and also the panelists. But as always, if you're going to use that, please be kind to everyone and only say, excuse me, nice things. As always, I want to remind you that NISS is the sponsor of these meetups. It's a nonprofit national research institute for statistics. It does many things, but its goal is mostly to promote statistical research and awareness of statistics. It does that through postdocs, conferences, and workshops, independent research, and the affiliates program of which Merck is part. You can get a listing of all their events if you go to www.niss.org. One I'd like to point out that might be of interest to this group is the 13th Annual Conference on Statistical Issues in Clinical Trials is a NISS-sponsored event in late April. It's going to be at the University of Pennsylvania, and the topic this year is around clustered randomized clinical trials, challenges and opportunities. And you can see there's quite an illustrious set of speakers that are going to talk there. So if you're interested, go to the NISS website for more information. This is, I don't know exactly how many NISS-Merck meetups we've had previously, but it's been close to 10 or so. The videos and slides for all those are at the NISS website. So if you have any that you're interested in, but you happen to miss, please go there and take a look. And an advertisement for the next one is going to be on Bayesian statistics sometime in the second quarter of this year. But for today, the topic is adaptive trials for drug development. And as I mentioned, we have a really great set of speakers that have a tremendous amount of expertise on adaptive trials. We're going to start off with Vladimir Dragulin talking about thoughts on the ICH E20 expert working group on adaptive clinical trials. And then we'll move to Kiki Deng, who's going to talk about two-stage adaptive designs in dose ranging studies and the alternatives. And we'll end with Scott Berry talking about master protocols and platform trials. So with that, I'm going to stop sharing and Vlad can start his sharing. And I'll introduce Vlad as we do that. So Vlad Dragulin is a vice president and scientific fellow in quantitative sciences at Janssen Pharmaceutical Companies, which is affiliated with Johnson & Johnson. He's the head of quantitative sciences consulting and is chairing the Q-Steps advisory committee. He joined Janssen in 2014 as the head of Adaptive Clinical Trial Center of Excellence, but he's also worked at GlaxoSmithKline, Wyeth, Pfizer, Quintiles, and Aptiv Solutions. He's a well-known adaptive design expert who's been in the field for 30 years or more. And he's the pharma rep on the ICH E9 R1 expert working group and on the ICH E20 informal working group, which he's going to talk to us about now. So with that, take away, Vlad.
Thank you very much, Dan, for introduction, and good morning, good afternoon, or good evening for everybody who is online. It's a great pleasure to have this opportunity to share with you some information about this ICH E20 now is expert working group, but at the time when I committed to present, I was member of so-called informal working group. So here is the outline of my presentation. First, I will explain why there was a decision to come up with a proposal for developing an ICH guidance on adaptive clinical trials. And I will give you a bit more details about the composition of working group. The process is first you put an informal working group, and then the assembly is approving that informal working group, and it become an expert working group. And then I will share with you some details of the first meeting that we did have in Singapore. So here is the background to these proposals. At least in the industry, there is an agreement and appreciation for the potential benefit of adaptive clinical trials. So this potential benefit is expressed first by increased efficiency of adaptive clinical trials. That means to save resources and accelerate market approval by mitigating the risk associated with, and I have several building blocks here, with the right dose, right patients, shorter study, and right sample size. Let's take one by one. So related to right dose, this means that this adaptive design or adaptive clinical trials provide us opportunity to do better dose selection and characterization of the dose response profile. And of course, ethically, it minimizes exposure of patients to ineffective toxic drugs. For the patient population, right population, this means that these designs allow to target the population or perhaps the subpopulation who benefits the most. There are type of adaptive design that provide that opportunity. Uncertainty around treatment effect can be mitigated by stopping early for superior efficacy or futility, and by that, of course, we can achieve shorter study. But also uncertainty around design assumptions, let's say treatment effect size, variability, so that can be achieved through sample size determination or sample size re-estimation, so-called type of designs. That was the first. Second one is about increased productivity. So adaptive clinical trials can better leverage information or data and reduce time at each stage by more efficient and informed design program in late development, optimizing treatment assignment to population, subgroup, or indication. The variations are enrichment design, response adaptive randomization, and so on. Combining multiple stages into a seamless design, for example, proof of concept and dose finding in a single protocol, in a single study. Better use of operational resources when combining multiple indications or compounds into a single master protocol. My colleagues, which will follow after me, will give you a more precise or more detailed example. For example, Kiki will probably explain some new developments in combining these multiple stages in a seamless design, and Scott will give us more details on the master protocols. And the last but not the least benefit or potential benefit of adaptive clinical trial is this increased portfolio value. That means to make the correct decisions at the earliest time point in the most efficient manner possible by enabling better management of resources across pipelines of products, maximizing the probability of success, and optimizing investment decision making. So to summarize, at least from the industry perspective, we see the potential benefits in this many kind of dimensions or opportunities. So, but of course, this comes with some challenges. And the first challenge that is highlighted is uncertainties that are present due to differences in current regulatory authority guidances with regard to the terminology of adaptive design, the lack of common principles for the design, conduct, analysis, and interpretation of adaptive clinical trials, and the lack of common expectations for documentation to support regulatory review. Second one is different perspectives among health authorities or regulatory agencies in different regions have resulted in uncertainty in the use of adaptive clinical trials in a global environment or in the global development program. So with these challenges, of course, we believe that there are still some opportunities, and that one of the opportunity is this harmonized perspective of adaptive clinical trials among the different ICH regions will allow sponsors and regulators to build an efficient multi-regional prospective plan for drug development which incorporates this innovative design. So this is why the pharma, as a professional organization of pharmaceutical companies in United States, together or with the support from FDA and from FPA, which is a similar association of pharmaceutical companies from Europe, made a proposal to develop an ICH harmonization guideline on adaptive clinical trials. So then what is the proposal for this and is now accepted as E20 guideline, ICH E20 guideline, and one of the latest that is under development today. The scope of this is a new ICH guideline on the design, conduct, analysis, and interpretation of adaptive clinical trials. So like I mentioned, the objective is then to provide a transparent and harmonized set of principles for the design, conduct, analysis, and interpretation of adaptive clinical trials. Although you know that ICH guidelines are mainly focused for confirmatory clinical trials, from the very beginning, we mentioned that that will remain the primary focus, but we expect the guideline to cover not only, let's say, phase 3 pivotal studies, but also some exploratory clinical trials or studies which have adaptive designs. So then an informal working group for this development of this guidance, there are some restrictions on how that informal working group should be created. And there is a protocol or a process in the ICH assembly that this group should consist of ICH members or observers. The expertise should be a balance of clinical and statistical experts with experience in innovative clinical trial approaches. And this ICH informal working group with the main scope to develop the concept paper and business plan was launched in June 2019. The members of both informal working group and then expert working group, I will explain a bit later what is the difference, is that different ICH members or observers are submitting candidates in order to join these groups, and then there is a kind of a selection committee which select the members of this working group. So the informal working group worked offline through bi-monthly teleconferences, and we worked together to develop the concept paper and business plan in order to be submitted to the ICH assembly meeting in Singapore in November 2019. And it was submitted, and ICH endorsed not only the concept paper and business plan, but also the creation of the E20 now expert working group. The roles are slightly different. It's not necessary that everybody in the informal working group will become expert working group and vice versa. It might be that there are additional members of expert working group who were not part of informal working group. So I have here a picture of informal working group, and then this informal working group was approved to become in full kind of content the expert working group, and we met in Singapore during November 17, 20th last year. And you can see here that because that was an initiative to develop the ICH coming from pharma, according to the process of ICH, the rapporteur is a pharma representative, and we have John Zhang being the rapporteur, and then always there is a regulatory chair, and for this expert working group, regulatory chair is Gregory Levin from FDA. You may know that Greg, together with John Scott, which is also a member of working group, they were primary authors of FDA guidance, draft guidance for adaptive design released in 2018, and which was finally approved now as a guidance last month. So then there are representative, two from each of the professional organization or representative of industry. You can see here pharma, this is for US, FPA, this is for Europe, Bio, this is now cross Europe and US, a new member of ICH is the associations of biotech companies, and there are two representatives on those. GPMA, this is the kind of industry in Japan. This one is from IGBA, I don't know the abbreviation, but this is India. And then we have FPMA, this is like China pharmaceutical industry. And then, of course, we have representation, not necessarily two, some are only one, from regulatory authorities across the globe. So FDA represented, European Community, PMDA or Japan, Health Canada, Swiss Medic, Anvisa, HS Anvisa is Brazil, this is Singapore, MFDS is China, and NMPA, this is China, Taipei. And then there are observers, and this is like Hong Kong and Saudi Arabia. So you can see here is a representation of all, both industry representatives and regulatory authorities. So progress made up to this point, like I mentioned, we met in Singapore and we reviewed regional regulatory guidances from Europe, US, China. China is in the draft format, and then PMDA. PMDA doesn't have a guidance on adaptive design per se, but they presented their guidance on data monitoring committees, and we reviewed some of the aspects related to adaptive design in that one as well. And then we reviewed industry perspectives on this because it's kind of a two-party type of enterprise. And then we discussed the scope of the future ICH E20 guideline, along with some specifics on definition of adaptive clinical trials, what should and should not be in the scope of the guideline, and table of contents. So we discussed a plan to move then from the scope to the future guideline document. We agreed to create a so-called writing team subgroups. And we achieved consensus on the working plan. We made a request to the assembly. We presented in the third day of our meeting to the assembly, where representatives from all these parties are present through their management committee membership. And they endorsed our concept paper and business plan. They endorsed our formation of the expert working group and work plan endorsement going forward. So that's what we achieved, and we are satisfied that everything was endorsed with no complaint, no other concerns. So just to go a bit more in details on the discussion, and you may ask yourself, why do we need ICH when we have so many already guidances? And I think that we reviewed this four, like I mentioned, definitions or existing definitions of adaptive design. And in this slide, in italic, I highlighted some of the aspects of the definition or some components of the definition that we highlighted that there is still inconsistency, and it's good to have harmonization. So first, of course, is you see here pharma working group publication with this definition, and I highlighted here the adaptive design uses accumulating data from the ongoing trial. Now, if you look in what China was proposing, they were saying accumulating data from the trial, but also external data. And it was a debate because that was in contradiction with pharma guidance, where they say adaptive design, if any design which is using only external data, it's not an adaptive design. So we went in the round of debates, what should be this harmonized definition to take into account this. And you can imagine that the push for use of external data is now in far front, because many sponsors are proposing now more frequently to use historical data, borrowing information from external data, real world evidence, and so on. So I think that it still remain one of the issues that should be resolved in this new guidance. Another one is about, for example, in EMA reflection paper, they definitely said that you have to satisfy full control of the type 1 error. It's not that others don't say that. For example, industry was using validity in order to capture that. But I think that it was a disconnect between, do we just need to focus on only type 1 error control, or more broadly in, let's say, focusing on false decisions? And I think that, those were then bringing us into the discussion about what should be the core principles then that should be reflected in the guidance. And you can see here that on the latest issue that I just mentioned, we are now will be proposing that it should not be just focus on control of type 1 error probability, but on control of chance of erroneous conclusions. That might also take into consideration perhaps type 2 errors, or even in, let's say, those findings when we don't have type 1 error, what are the erroneous conclusions and how we capture those as a principle. The other one is not focusing just on a control of type 1 error through the methodology of hypothesis testing. Also, we need to pay attention on reliability of estimation. It's well known that although there are known and acceptable methodology for how to deal with the adjustments, required adjustment for controlling type 1 error through this, let's say, p-value combination methodology or others, but estimation at the end of this type of design is not yet completely sorted out. So a core principle then will be there that we need to make sure that we have harmonization on what we mean by reliability of estimation. A maintenance of trial integrity is another principle that should be highlighted in the guidance, and then that is linked or interlinked with adequate trial planning. In FDA definition, you can see here that they say is prospectively planned. So this means the need for this before the study, not ad hoc reaction to the data, should be a principle. And because I was member of a pharma working group on adaptive design, from the very beginning, we highlighted that adaptive should be adaptive by design, not ad hoc, and is not a protocol amendment. And I think that you can see that there is kind of a commonality there, but it will be good to put it in the ICH guidance, to be clear about that. It was a lot of debate about another principle that some of our regulatory colleagues were trying to put as the fifth one, and it's coming under the kind of term, design is fit for purpose. So let me explain what that means. That they are trying to say that as a sponsor, when you propose a protocol for adaptive design, that in the recommendation, you also have to show why did you choose this type of adaptive design by not another one. And probably many of my colleagues from industry have this experience that in the past, when we were proposing some adaptive design, you will receive back recommendation, why didn't you use group sequential design? So I think that we would like to have a bit more time to discuss and come perhaps with a compromise that it doesn't need to go up to the recommendation why one design versus another is selected. As long as design satisfies the core principle, that design should be appropriate and should be accepted. But this is still under discussion. So this is the draft structure of the guideline that we agreed upon, but of course there are a lot of work to be done. So you can see it will be introduction, benefits, rationale, and scope, and then definition and concepts, key principles, including guidance on how to achieve those principles, will be the core of the document. Adaptive trial landscaping and relevance, and then maybe some details on special topics. And here is the list. Of course, it's not exhaustive. Maybe some of this will be excluded in the final version, but these are just highlights that I want to share with you. Another one, which is also very important, about documentation and interactions with regulatory authorities, that we believe it should be a harmonization in this guidance, and perhaps even including some examples. From my experience on E9 addendum, I think that we were advised and we agreed that it is not good practice in guidelines to provide examples. There is a risk then that that example will be interpreted that that is a rule, but also probably we will be able to develop training materials on this ICH guideline as a follow-up, and that may be highlighted or emphasized, some of these more technical details in the example, but maybe not in the guideline. But we didn't agree, and we don't have final conclusion on that. So this is the future key milestones. This is an enterprise which is not going to be done quickly. We are serious about that, and you can see that you would expect to see the first step one, sorry, for comments only in November 2021. And then, of course, we receive submitted comments from the parties, and we have to incorporate those, and then we come up with so-called step 4. Back in my experience with E9, we received about more than 1,000 comments, and we had to spend a lot of time how to address all those comments and recommendation, very useful ones, in the final revision. So this is our kind of roadmap. And in conclusion, I want to report to you that this E20 Informal Expert Working Group collaborates well together. We made a progress on the details of the scope. We also highlighted some topics needing further discussion, and no major barriers to harmonization have been identified so far. And the progress of the working group, according to our work plan, is on track. With that, I will close here and give the forum, the floor back to Dan.
Great, Vlad. That was great because, you know, many of us know that this sort of stuff is going on, but don't really have an idea of what the issues are and what the end product is likely to be. And so that was a great primer on what's going on, what the issues are, and where we hope to be in the future. So with that, let me introduce our next speaker. Kiki Deng is the Senior Principal Biostatistician, excuse me, is a Senior Principal Biostatistician at Boehringer Ingelheim Pharmaceutical. She is currently a member of the methodology expert team within Global Statistics, which focuses on statistical methodology and innovation. Her research areas include hypothesis testing and modeling and dose finding, pragmatic considerations in designing trials, and adaptive design aspects of those trials. Before she joined the methodology group, she served as a lead statistician for multiple projects across different clinical development phases and different therapeutic areas. She holds a bachelor's degree from Peking University in China and obtained her PhD from the University of Minnesota. So with that, Kiki, we're looking forward to your presentation.
Thank you very much, Dan, for the nice introduction. And also thank you very much for inviting me to this webinar. And hello, everybody. I'm very happy that today I have the opportunity to talk to you about some of the work we do for the adaptive design in dose ranging studies. So for those who works in oncology, the dose finding process is usually conducted in phase 1 dose escalation studies. So for phase 2, typically there is no dose ranging studies per se. So for today, my talk is going to focus on the non-oncology background. For non-oncology development, and especially in chronic disease, we know that there are usually 2 key objectives for phase 2. The first one is to demonstrate the drug works, which is called proof of concept. And then the second one is to find out the appropriate dose for phase 3 confirmatory studies. So usually for the phase 2 development, there will be 2 studies. The first one is called phase 2A study, usually with 2 arms, a small study. The dose will be a high dose close to MTD, maximum tolerable dose, or maximum feasible dose to maximize the chance to detecting the efficacy signals. And then once the concept is proven, usually it's followed by a moderate-sized phase 2B study. We usually have a dose ranging study there, and there are usually 3 to 5 different active doses plus placebo. So in recent years, we see a very strong emerging trend from the industry that people tend to combine the proof of concept and also the dose ranging into one single trial, as you can see from the figure at the left. It could be the sample size could be equally allocated to different dose groups, or it could be having unequal allocation with slightly more samples on the 2 sides and have less samples in the middle. The reason is usually that kind of allocation ratio increases the precision of the estimation of the curve and also to increase the power under the fixed total sample size. So, of course, the advantage of this combined approach is to shorten the development timeline and also that lead to a smaller total investment if the drug works. However, it also comes with a cost or risk that if the drug doesn't work, then there will be a higher sunken cost. So in order to mitigate the risk in this combined approach, there are usually 2 ways to handle it. The first one is just following similar idea as a traditional development path to have 2 separate or even 3 separate different studies. Instead of the typical proof of concept studies usually people use, there is a concept called early signal of efficacy proposed by people from Pfizer. So this ESOE studies usually have a small sample size with a relatively large type 1 error level, for example, 40%, as a start of the development. And then the other way to mitigate the risk is to have adaptive design and look into the interim data and then accordingly adapt the decision afterwards. So one of the commonly used adaptive design is to have an interim futility analysis. Next, I'm going to use an example to compare some of the popular and available approaches out there. This is a hypothetical example, but it's very typically seen in real practice. So suppose we have a continuous endpoint with 4 different active doses against the placebo, and I also put in some typical assumption about effect size, variability, type 1 error, power, and we look into different candidate model shapes for the dose response curve. In terms of the statistical methodology we look into, we use either highest dose against the placebo for the hypothesis testing that the drug works, or we use the multiple comparison procedure and modeling approach. This is called MCP-mod. MCP-mod is a very popular method nowadays for dose finding studies. for the sake of time, I'm not going to get into details about this approach, but in the high level, it is a methodology that combines the data from different dose groups to test whether there is a non-flat dose-response relationship. So the benefit for using that is usually it's very powerful, and also it's robust to different dose-response curve assumptions. In addition, because the allocation ratio is important in terms of the final performance, we, in order to have a fair comparison, we match the overall allocation ratio to be 2 to 1 to 1 to 1 to 2 across the 5 different arms. First, the first option we look into is a traditional way to have 2 separate trials, but we follow the early signal of efficacy concept for the first study to have a relatively small one with a higher type 1 error rate of 40%. And then if there is a signal detected, we move on to a full dose ranging studies with a type 1 error of 0.125, and it's based on MCP mod. So from the project level, combining these 2 studies, we can see the overall type 1 error for the development program is controlled at 5%. And we use 95% of power for each study. Therefore, overall power is 90%, as you can see in the bottom calculation. So in addition to that, the average sample size can be straightforwardly calculated under non and alternative hypothesis. The second option we look into is to have a futility analysis in the middle of the trial. We consider 2 designs. The first design is more similar to the typical MCP mod study with one combined with one single study for the 2 purpose. And we just simply add a futility analysis in the interim. And then for the right panel, you can see this is what we call the stepwise design. So the idea is to have a high dose, a single dose against the placebo for the first stage. It's only that when the signal is there, then we expand to enroll the other dose groups. The reason we do that is for the lower and middle doses, usually there is a possibility that they don't have effect or they have a very small effect size, but it's not certain about it. Therefore, having those arms usually dilute the signal of efficacy, and that's reflected on the sample size needed here. Basically, we match the overall type 1 error to be 5% and overall power to be 90%. We also match the interim stopping probabilities to be 40% under none. So in order to do that, actually you need a much larger sample size for the first stage on the left, which is close to maybe 230, and on the right to do the same to gain the same probability, you only need 130 patients. In summary, we look into 5 different approaches, and we actually we actually match the type 1 error power and the stopping probabilities in the interim. The table 2 summarizes the sample size needed for each options. So because we match all the probabilities, then the smaller sample size needed means the design is more efficient. The first 2 columns represent the 2 traditional approach. The first one is to have 2 separate trials. You can see that this is the most conservative approach that's reflected by the maximal sample size and also the average sample size under alternative hypothesis, that both are the largest among all the different approaches. However, this have the advantage that when the drug doesn't work, then you have a smaller sample size and smaller sunken cost. In comparison, for the combined approach of having one single trial, you do get some benefit of having a smaller maximal sample size and the smaller sample size when the drug works. However, it comes with a cost that when the drug doesn't work, then you have a higher sunken cost. That's the highest among all the approaches. So for the three adaptive design, the third one, the fourth one are the two represented. I presented in the previous slides. And the last approach is just very similar to the fourth approach. The only difference is we use two sample comparison in the final analysis instead of MCP mod. So it's slightly less powerful. Comparing those adaptive designs to the two other options, you can see the stepwise MCP mod performs best in terms of it have a smallest average sample size when the drug doesn't work, even smaller than the two trial approach. And then when the drug works, actually it have 100 patient less so basically 100 patient reduction comparing to the two trial approach. It is slightly larger than the single trial approach, but I think overall it strikes very nice balance between the pros and the cons of the two traditional approach. So the next slide is just a graphic presentation of the numbers that's shown in the previous slides. And you can see the reduction here when the drug doesn't work, and also the comparable performance when the drug works. Hopefully by these results, I can demonstrate to you that having the two there are some benefits of using adaptive design and having the highest dose against the placebo as a first stage comparison. You do have some benefit here in terms of the efficiency. Okay. So next, I'm going to switch gear a little bit to talk about one alternatives comparing to what I introduced so far. So we call this a two-stage stepwise design. The idea is similarly that we also start with highest dose against the placebo. For example, we can enroll 30 patients each arm. Then after at the interim analysis, you can look at the treatment effect size. If it's promising and higher than the threshold, you can expand the study to enroll additional doses. We call it go fast mode. On the other hand, if the interim signal is not that good, then you continue to enroll additional 30 patients on the two doses, but keep the current study as a two-arm study for proof of concept. And then at the end of the study, you can make a decision whether you want to move on with the development or not. So we call this go slow mode. As a summary, this approach starts with two group comparison with high dose against the placebo, and then depends on the interim data, you choose a path for the next development, either go fast or go slow. In terms of how we calculated the sample size to set up the trial, for the go slow mode, it's basically the same as you would do for traditional proof of concept studies. And for go fast mode, it's basically the same as you would do for dose ranging studies. We use the same delta standardized effect size, the same power. The only difference is that we recommend a slightly higher type on error if you move on to go slow mode. The reason is because if you succeed this trial, the next trial will be a moderate-sized dose ranging study instead of a large-scale confirmatory phase 3 studies. Therefore, the level of tolerance for the risk could be slightly higher. So the basic idea for this design is coming up from the 2-in-1 design that Kong Chen and his colleagues from Merck has proposed in the oncology phase 2 and 3 seamless design. However, in oncology, you don't really have this dose-finding component in phase 2. Therefore, you can think about our proposal as a modification of the similar concept to a non-oncology phase 2A and 2B combined study. Therefore, we can borrow the way and from mathematical point of view to demonstrate that actually the probability of continue the development when the drug doesn't work can be controlled at the level of the bigger alpha level of the two development paths. So the bigger value of alpha tilde and alpha. So this graph summarizes some of the important aspects of the operating characteristics of the design. The x-axis is a cutoff or threshold for interim effect size, and then the y-axis is different probabilities. One of the very important or interesting properties we find is that under this kind of design, the probability of continued development is well controlled at 88%, regardless of the cutoff you're using. This is very different from a futility analysis where if you increase the threshold for the interim decision making, then the power will have a sharp drop. Actually, the power of a futility design is basically shown by this probability of go fast and succeed, which is this dotted line. And as expected, this will decrease sharply when you increase the interim threshold. That's part of the reason when people use futility analysis, they cannot or they tend to go to relatively small cutoff values. Sometimes even you didn't observe some effects, any effect size, or observe a very small effect size, you would still continue the trial. The reason is to protect the power under a given sample size. So in this design, we don't have to worry too much about that. When you increase the cutoff threshold, you do have a problem that the probability of go slow and succeed will increase. This is sort of have a reversed results. However, although it's not very desirable to have this kind of results, however, it is much better than missing a good drug or kill a good drug. So this is one important property of the trial. We also look into the recommendations on the interim cutoff. We look into the expected sample size for this kind of design. Here, I want to point it out that in addition to the current sample size, it's important to consider a penalty term for the case where you move on to the go slow mode but succeed in the end. Because in that case, you have to conduct a second dose ranging, another dose ranging studies to find out to the appropriate dose. Therefore, there will be a penalty for that kind of situation. And based on that, you can find some sort of a sweet point in terms of the threshold values. It's approximately slightly higher than half of the expected effect size. So with a region here, you can obtain most of the benefit that you would have in terms of the sample size reduction when the drug doesn't work. At the same time, you actually didn't increase the sample size too much when the drug does work. Okay, so in summary, for the proposal I introduced here, there are two key factors. The first one is to start with a high dose against a placebo and then expand if the interim data is promising. As I introduced at the beginning and also hopefully shown in the simulation study, that this is usually more efficient to generate a meaningful signal with a small interim sample size. And then the second one is the two-in-one concept instead of the futility analysis concept. First of all, the probability of continued development is very stable under either path. The only difference is you're going to move on fast or slow. And then because of that, you can actually choose a more meaningful interim boundaries for decision making. One of the advantages I haven't discussed so far is this approach also naturally takes care of the patient overflow. Patient overflow or pipeline patient is a typical problem in the adaptive design. That when I see that, it means when you have the interim analysis, actually there will be, at the time of the interim analysis, there will be some patient who they start the treatment, however, they haven't reached the primary endpoint yet. So for those patients, in futility analysis, the data is not formally incorporated in the final decision making if you decided to stop the trial. However, with this design, under go slow mode, this part of data is still taken care of in the final decision making. So that's one of the advantage for this design. I also want to point it out that the interim message for this kind of design will be very different comparing to futility analysis. In futility analysis, if you stop the trial, if the data didn't pass the interim threshold, then your message would be, I'm going to stop the trial for futility or for lack of efficacy. But for this two-in-one concept-based design, when you didn't pass the threshold, you would see we decided not to expand right now, but the final decision will be made at the final analysis when the data is complete. So that's a much more neutral message than the futility analysis. And then also, this kind of design can be extended to different variations. For those finding study, you can either use MCP mod or two-group comparison. For MCP mod, it's taking care of the power advantage. You can, for two-group comparison, you can take advantage of the group sequential design for its simplicity. There are other approaches there for trend test. For example, those can be naturally incorporated. And in addition, you can use a short-term intermediate endpoint for interim analysis. The type 1 error will be similarly protected. However, the performance, of course, depends on how the intermediate endpoint correlates with the final endpoint of interest. At last, although I mentioned there are some there are a lot of advantage of having adaptive design, but there are also, on the other hand, some advantage of having two separate trials, especially operationally. They could lead to smaller initial commitment of the resources, and also you can have some flexibility in terms of how much data and what type of data to be collected. They can be different from the two trials. The study duration can be even different, and the endpoint could be different. Okay. So finally, I want to acknowledge some of the peoples for this work. I want to thank Nai-T and Ting and Xiaofei Bai from Boehringer Ingelheim for co-authoring the paper. And I want to thank Yutao Liu from Columbia University for conducting the simulation study I demonstrated in the first part of the work. And I also want to thank Kong Chen from Merck for the discussion of the two-in-one design. So there are some references, and as mentioned earlier, we will leave the questions to the end of the webinar after Scott's talk, but thank you very much for your attention. Then I will hand it over back to Dan.
Great. Thank you, Kiki. That was a wonderful explanation of these two-stage adaptive designs, and I'm sure the attendees really appreciated it. I want to just, before I introduce Scott, and while he's getting his slides up, remind folks that even though we will address questions at the end, you can start putting the questions in the Q&A now so that we can get sort of prepared for that. So please click on the Q&A button and put your questions in there if you have them. And with that, let me introduce Scott Berry. He's the president and a senior statistical scientist at Berry Consultants. He earned his master's and PhD from Carnegie Mellon and was assistant professor at Texas A&M before co-founding Berry Consultants in 2000. Since 2000, he's been involved in the design of hundreds of Bayesian adaptive clinical trials in the pharmaceutical and medical device fields, and has become an opinion leader in the field of Bayesian adaptive clinical trials. He's a primary designer of commercial software used in the simulation and construction of innovative Bayesian adaptive designs. His research interests include Bayesian methods in clinical trials, adaptive clinical trial design, Bayesian computation, meta-analysis, and hierarchical modeling. He has more than 30 refereed publications in a lot of the most important statistical journals, and co-author of a preeminent book in adaptive designs, Bayesian adaptive methods for clinical trials. And so with that, we look forward to your talk, Scott.
All right, thank you very much, Dan. Thanks for the intro, and appreciate the invite. Thanks everybody for joining. Hi, so I'm going to talk. I suppose I could move my slides here. Sorry. So I want to take a step back, and according to the internet, which we know the internet is never wrong. The first randomized human trial was designed by Austin Bradford Hill. And so we'll consider this, and this I'll show you a little bit from this paper. It was a randomized trial of streptomycin for the treatment of tuberculosis. And the results came out in a 1948 paper. And so here are results from this trial. It has a traditional table 1 and table 2. Looks very similar to what you would see now from the results of a randomized trial. For streptomycin was very effective. An interesting aspect of this is they had an ordinal endpoint for the clinical outcome of individuals. And so even 70 years ago, they talked about how to analyze an ordinal endpoint, something we still discuss quite a bit now. But the other part to this, and there's the interesting part that there's a typo in the 1948 paper. They make a mistake in dividing 4 by 55. But the trial design hasn't changed much in 60 years. Randomization was an incredible innovation. We left the anecdotal medical research, and it was an incredible innovation, but the trial design hasn't changed much in 60 years, 70 years from the time of this publication. I think things have been happening in the trial design within, say, the last 10 years. So the comment is now that the clinical trial science itself is being innovated. We have incredible medical therapies in the last 60 or 70 years. If you look at the innovation in the therapies themselves, it's stunning, the innovations that have been made in that area, even if the innovation in the trial design science hasn't changed much. So now we're entering into this innovation in the trial design. So I'll present in that sort of a way. So I'll discuss a little bit, and Vlad talked about the adaptive design guidance. I'll pull a few snippets out of that, and particularly parts of it that I like. and the draft guidance on complex innovative designs. I'll show you a couple quick examples, and these are nice examples in the fact that they're done, and a great deal of it is public, and you can see the final result of some of these. So they have happened. Complex innovative designs has happened. And then I'll move on and say a little bit about platform trials and show you an example of that and how that's really a confluence of many of these things. And it's in line with this idea of clinical trial science moving forward. And what's beyond that? And how does that play in? Okay, so the two key guidances that I'll just present some snippets on are the guidance on adaptive designs, the draft guidance on complex innovative trial designs from the US FDA. My overall summary of some of this, and I'll assume you've read these, so I'm not going to go through and lay out what they do, what they don't do. I think the most important part of both of these is that they exist. It takes down a huge barrier or perceived barrier that some may have had. Oh, the FDA doesn't like this. Oh, they're not well understood. And whomever it was who may have provided barriers, it's really hard to take that position. I don't think either of these guidances are limiting. The adaptive design guidance says at the very beginning, this is no way limiting. And there may be cautionary statements, but I don't think they're limiting, and I think that's huge. The 2010 draft guidance were perceived by some to be limiting, and they could say, oh, see, FDA maybe doesn't like this. I think that's gone. I think it completely opens the concept that adaptive designs and complex trials are good, where beforehand, maybe not. But now I think it paints it in a positive way. The FDA, in some ways, is leading this innovation. I think they're forward-looking. And a danger of guidances, and I really enjoyed Vlad's presentation of the ICH discussion, and the danger of some of this is it may be appropriate today, but restricts innovation tomorrow. And I don't think these guidances do that. So this is my overall summary of that. Let me move this picture of myself, which is a little bit weird to look at. Okay, so some of the snippets, and I added the color, otherwise these are pulled directly from the guidances. These are the adaptive design guidances. In still other cases, such as many Bayesian adaptive designs, it may be critical to use simulations to evaluate the chance of erroneous conclusions. Vlad talked about this term erroneous conclusions. In some ways, what's really nice about that is it's not a focus on type 1 error. Type 1 error is one kind of erroneous conclusion. In some settings, as in Bayesian trials where you're using prior information, type 1 error may not be the relevant conclusion to worry about in those settings. It also brings out that simulations play a critical role. These are two huge moves in the last 10 years, both of these, that the presentation, first of all, that simulations are appropriate and critical, and this idea that there are lots of conclusions to worry about, and it really, I think the adaptive design guidance moves away from type 1 errors being the be-all, end-all. Which are really important moves forward. The scale is relatively unimportant. This is whether it's frequentist, Bayesian, the scale isn't as important as the operating characteristic of the design. Does it get the right answer? Does it measure things appropriate? Is it efficient? Is more important than is it Bayesian? Is it frequentist? And I think that's a critical recognition. moved off myself. It opens up response adaptive randomization. It talks about that there's been controversy on response adaptive randomization, but it absolutely lays it out there as something that might be appropriate in a design within the setting. It also recognizes that you might do 50 interim analyses where you do response adaptive randomization. It doesn't mean you inflate type 1 error. I think the adaptive design guidance does a nice job of separating this concept of an interim analysis from the action taken there. In group sequential designs, it's become this idea that if you do an interim, it causes a penalty and an inflation. And this moves away from this idea, which certainly isn't true. There are many interims that don't inflate type 1 error. Futility, as Kiki discussed, doesn't inflate type 1 error. Response adaptive randomization doesn't inflate type 1 error. And I'll show you a quick example of this. And so I thought that that was a really nice aspect of the guidance. It opens up this notion of controlling type 1 error through simulations. I think this is a huge move forward in clinical trial science. At some point 10 years ago, 15 years ago, there was this notion that if you couldn't show analytical control of type 1 error, it wasn't appropriate design. It's an incredibly limiting thing for a clinical trial that you have to have pencil and paper understanding of the behavior of your design. Imagine if bridges and airplanes and cars, you had to have analytical understanding of the aerodynamic of the car. We wouldn't be where we are today, and clinical trial science needs this same aspect, that the designs of today and tomorrow will be much more complicated, and they will be built on simulation. They will be understood on simulation. Computer-aided design within this aspect of it, and the adaptive design guidance understands and presents this. It also opens up, it's kind of interesting, this quote, that there may be simulation after the design is over to understand whether the original simulations were appropriate. We did a grid search of values. We looked at the operating characteristics. There can be post-simulation to understand what actually happened and did we simulate it appropriately. That's not true of every design, but I think some designs maybe this is quite appropriate, and this opens up this idea. The same principles apply to Bayesian adaptive designs and non-Bayesian. It takes this agnostic view that Bayesian is not some foreign thing. Again, it's about the operating characteristics, getting the right answers, about drug effectiveness. You might do a design with adaptive features that are Bayesian and have a frequentist final analysis. That can happen in trials, and I'll actually show you one like that coming up. The complex innovative designs, the draft guidance, what's really nice again is its existence. And this is really, there's a lot of forward-thinking aspects of complex innovative designs. And even in the definition of what it is, although CID has been referred to as complex, adaptive, Bayesian, and other novel clinical trial designs, there's no fixed definition of CID because what is considered innovative or novel can change over time. For the purposes of this guidance, CID includes trial designs that have rarely or never been used. I love the aspect that they present this, that in a guidance, you don't want to list what's okay, because a year from now, 5 years from now, we should have much better designs. And it opens up that we need to understand designs that have never been used as situations and clinical scenarios 5 years from now may need that. And so this idea that you have to do something that's been done before is the opposite of innovation in some way. So I love that aspect of the CID draft guidance. It does present that it's a common feature now, at least, that CIDs, the need for simulations in some ways, presents what is complex. Rather than mathematical formula, pencil and paper solutions, I added that, to estimate trial operating characteristics. So that a lot of times is the definition of complex. I'll use that as a definition of complex, that we have to use simulation to understand the design. And it presents this idea of a simulation report as a new thing in clinical trials that's needed for complex adaptive trials. So these are great snippets that I thought were important in these. So I'll present a couple example trials and explain how they sort of fit into all of this. I won't show the details. There are publications on both of these trials. You can look at the details of them. This was the diabetes phase 2/3 seamless trial, Eli Lilly trial, for their drug, Trulicity. It was a phase 2 trial with 7 doses, a placebo and an active comparator in the phase 2 portion. There was a burn-in period of equal randomization. Then there was a period of response adaptive randomization. Every 2 weeks in the trial, the response adaptive randomization was updated. The RAR was based on 4 endpoints: HbA1c change, weight loss, blood pressure, and heart rate, combined in a weight function or a utility function to weight the response adaptive randomization. At some point during this stage of the trial, between 200 and 400 in phase 2, the trial could move to phase 3. It was done by algorithm, and it would select 1 or 2 doses to move forward into the phase 3 portion of the trial. It also would spawn other phase 3 trial, like a cardiovascular risk trial. It could stop for futility, or as Kiki sort of referred to this as, we're done enrolling in phase 2. It's not so much futile as let's just make this a phase 2 trial, get the final results, and then make drug development decisions. I'll refer to this as futility, but she gave a better description of what that actually would be. If the trial went to phase 3, it would be fixed randomization with doses selected by an algorithm. It was not selected by a DSMB. It was actually in the charter that they couldn't change the algorithm. They only monitored the algorithm. And the algorithm could pick 1 or 2 doses. The size of phase 3 was adaptively selected with constraints. And it would be fixed randomization during the phase 3 portion if it happened. The final analysis, as I presented in the snippet, all of that was done with Bayesian utility functions and modeling to make those decisions. But the final analysis was an ANCOVA model with a threshold of 0.020 to control type 1 error. That threshold was entirely selected through simulation, a grid search over null scenarios to demonstrate control of type 1 error. Very consistent with this notion that we don't have analytical control, but we've done an adequate grid search for this. And there's a quote from the EMA during this aspect of it, that they've run out of reasons to say no, and they accepted this control, and this was 10 years ago, but consistent with the current US FDA guidance. I won't say much about the utility function. The modeling is there was a Bayesian repeated measures model, a longitudinal model over 4 endpoints, and a weighting of the value of these. And the safety endpoints could demonstrate that there was no value to these, that they were not safe during the course of it, as well as building predictive probability of phase 3 success for the different doses. The trial ran, and really the running of the actual trial was just something like 3 million and first time it ran. We ran it in silico many millions of times to make sure it was going to run exactly as we wanted it to, as Eli Lilly wanted it to. The trial ran at 200 patients at the first time possible. It moved to phase 3. That spawned additional phase 3 trials. It selected the 0.7 milligram and 1.5 milligram doses out of the 7 possible doses to move forward. You can read about the details of that. And it was approved based on the subsequent phase 3 trials. In 2014, interestingly, there were some projections on the drug itself might make 1.3 billion by 2020. By 2018, the drug had made $3.2 billion, and 2018 is one of the largest accruing drugs. And in many ways, the seamless phase 2-3 trial sped up the approval of that and saved money at the end of the patent life for a drug that's making multiple $1 billion a year. The other part of this is the alternative design was a typical 3-dose phase 2 trial. The doses selected for phase 3 that are now doing very well in the market may never have been looked at. So part of the story of this is the ability of an adaptive design to explore 7 doses, where a traditional design could have looked at 3 doses for the same time and resources. And it may never have picked this optimal dose. It was all very consistent with the draft guidances of a complex, innovative design. The Dawn trial, and I'll go very quickly over this, you can read through this. This was an adaptive enrichment trial of a device that allowed for enrichment during the course of the trial to make sure the thrombectomy device was being effective for all patients, and it could restrict enrollment. There was a concern in stroke devices that it might actually be harming some individuals to restore blood flow. The trial ran. It was designed by algorithm. Type 1 error was controlled by algorithm. It ran exactly as designed during the course of it. It stopped for expected success at the first point it was able to. It actually never enriched. It was demonstrating benefit for all patients. So it's kind of an interesting trial, a complex adaptive design with enrichment, stopping rules, futility rules that stopped at the earliest time point, never enriched, demonstrate very high success. You can read about the results. The whole role of the enrichment is somewhat subdued in the paper because it never enriched. In some ways, this is a really nice thing that despite a really complex design, if you look through the results, it's about the data. It's about the success of patients and what it says. It's not about the complexity of the design. The primary analysis was also Bayesian, so you can look at the results in the New England Journal of Medicine talking about posterior probability of superiority as well. So that's an interesting part of it. In the adaptive design guidance, it talks about platform trials. And so this is this aspect of all of this stuff coming together, adaptive designs, complex innovative designs, master protocols, 21st century cures. And there's another draft guidance from the FDA on platform trials and master protocols. All of this, here's an example of that, which is Precision Promise, which is a platform trial in pancreatic cancer. The sponsor of the trial is a patient organization, PanCan, that is sponsoring it. And the idea is multiple pharmaceutical companies would bring drugs, or sponsors would bring drugs into the trial, into a common trial. It itself is complex and innovative in the sense that it's entirely built by simulation. It has an aspect of response adaptive randomization over all the experimental arms. There are decisions made about each of the arms adaptively. It's a seamless phase 2-3 trial for each arm. It also does response adaptive randomization for the patients. There is re-randomization. If a patient progresses on an experimental therapy, they can be re-randomized to another experimental therapy in there. And the primary endpoint for this is overall survival. So now you might have a patient that has experimental drug 1, experimental drug 2, and then has an overall survival result. And the modeling has to figure out how much of that survival time is due to each of the drugs in a phase 3 setting. So this is these are simulations. This is the last slide of an example trial where 14 different drugs, actually 12 different experimental drugs go through. One is demonstrated to be superior in the trial. This is the adaptive randomization over time. There are 2 control arms, but there's adaptive randomization. So these are all of these things coming together in a platform trial with multiple drugs. So it has a seamless phase 2-3 aspect for each drug. It is adequate and well controlled for each drug in the trial. It has adaptations for patients. It's built by a patient organization. So it is this aspect that patients can move through and have adaptive allocation to arms when the previous arm isn't working. This all has to be done in a phase 3 setting. It has many aspects together that make it CID. The modeling of sequential therapies, common control arms over different time in the trial, response adaptive randomization, seamless phase 2-3 for each drug. It's entirely controlled by simulation of type 1 error. It's not analytically controlled. The entire thing is simulated. So this is, I think, moving adaptive designs, moving platform trials forward. So what's the future? I've thrown out embedded platform trials, merging clinical care and learning all at the same time. I hope we get there soon. But the beauty of this innovation and these guidances is it's not restricting. The trial design science innovation, I think, is going to be awesome in the next 10 to 20 years. I don't want to restrict it by saying what it can be, and I don't think the guidances do either. So it's only restricted by our imaginations. And while in many ways in clinical trial science tomorrow is here, it's pretty exciting what these guidances, I think, open up for where clinical trial science can go. So I will stop there and turn it back to Dan.
Great, Scott. Thank you. That was a great presentation of really some incredible things that are going on. So really appreciate that. I think we just have about one question that's been posed. So let's start with that. And then if the panelists have questions for each other, we can do those. And if not, then I'll ask some of my own. So the one question we have is for you, Kiki. Someone was asking that if you have an interim analysis and you have missing data and you're going to use a missing data technique like multiple imputation, does somehow the boundary change? Do you have to somehow take into account the fact that you're using a missing data technique, or only the completers used, or how is that handled?
Well, when there is missing data in the interim analysis, I would expect that the effective sample size actually drops. So it's not really, you're not really getting what you plan to get in terms of the sample size in the interim analysis. In practice, I would still probably comparing to the same efficacy boundaries if the number is not off too much. However, the operating characteristics like the different probabilities might be slightly off what you are planning to do at the beginning of the study. So this overall, I think the way you handle it might be very similar to the way you handle it for the final analysis. You could inflate the actual sample size a little bit to account for the dropout. But in practice, if you are not missing too much data, I think you can still follow the original design, but accept or evaluate the impact on the different probabilities.
Great, thank you. So again, I'll do as I just said. Did the panelists have any questions for each other before I dive in?
I have a question for Vlad about the new work that's planned for the expert group. So you, in your talk, you mentioned the overall structure of the upcoming papers that's being planned. I was wondering, are you expecting from the content point of view, it will have a lot of overlap with the FDA new guidance on adaptive design, or it have a very different focus comparing to that?
Thank you, Kiki. It's a great question. You see, ICH is supposed to harmonize all these guidances which are already present in some of the regulatory agencies. So in this case, FDA guidance on adaptive design is already a law, or is in and I think that what the intention will be of the ICH working group is to try to harmonize. Will the final ICH guidance be very similar to FDA guidance? It's to be decided. I think that we now have representation from PMDA, from China FDA, from Europe, and maybe they are not ready to say that what FDA developed is good for harmonization across the globe. And that is exactly the scope and the reason why this group was created, or working group was created, in order to come with a solution. So it looks like we do have very much agreement in the team that FDA guidance may be the core, but of course the final version is to be decided, and how much similarity will be in the ICH guidance versus FDA is to be still seen.
Thanks, Vlad. I think I agree with you that because of the alignment across the different regions and countries, we expect maybe the position or message might be slightly different. However, it seems like at least for the topics that will be covered in this new paper, there could be quite some overlap, right?
Yep. Yep.
Okay. Thank
Great.
you.
Thank you. And we really only have time for one more question. I want to ask Scott. So, I mean, you showed some great examples of successes of adaptive trials, but, you know, you've worked on a tremendous number of these sorts of trials. I'm curious, and I didn't expect you to show ones that had gone badly, but you must have experienced some that haven't gone so well. I'm just wondering what you see as the major pitfalls that folks can fall into if they try to design trials like this?
It's a good question. Yeah, I suspect that I wasn't going to show things that went wrong. So what does it mean to go wrong? We've done a number of trials where the trial stops for futility. And that in many ways is a success of the design. Hopefully it's a success of the design, that it efficiently got the right answer. And the design can't make the drug better, but can it find the right answer as efficiently as possible? So in many ways, I consider futility decisions typically to be really good trial results. Almost all of our examples of things going wrong. So to back up a little bit, you can design an adaptive trial that doesn't work well. The safeguard of that is simulations. It's so important to simulate and compare, and it should come out with enough simulation that the design might not work well in certain situations. But most of our stories of things not going well are operational. It's the data's not done appropriately, things are lagging, switches are not flipped appropriately, We have some examples of what I would say DSMBs going rogue a little bit in that, but almost all of it are operational things that make it a little bit sluggish or not go as designed.
Great, thank you. Okay, and with that, we've reached the end of our time. I want to first thank the speakers. I thought that was 3 just really excellent presentations, and certainly I learned a lot. And I'm sure that our attendees did as well. So we're really lucky to have such great expertise. And you know, to the audience, these folks are donating their time. So you know, if you see them at a conference or whatever, go up to them and thank them because it really is a service to the community. And lastly, I want to thank some other folks. Christy Trang-Stein from NISS helps me organize these. Glenn Johnson and Jim Rosenberger worked on the technical parts with the Zoom, so I want to thank them. And then remind folks that we'll hopefully see you again next quarter, probably in April, for our meetup on Bayesian statistics. So thank you for joining. I hope you got some great information out of it, and we look forward to seeing you in the future. Okay, we'll sign off. Thank you very much.
Bye, everybody.